Today is November 25th, 2018. Hundreds of the world's top molecular biologists, experts on the new CRISPR technology, bioethicists, scientific reporters, and policymakers are gathered at the University of Hong Kong for the second International Summit on Human Genome Engineering.
On the stage is Chinese researcher He Zhuanghui, one of those CRISPR experts who has his audience enraptured by the words he just said. Many passionately objected to his presentation, calling him irresponsible and reckless. What could Zhuanghui have said or done to have so intensely worked up this crowd of polished professionals and authorities?
In order to understand what happened at this now infamous convention, we need to rewind all the way back to the discovery of the revolutionary technology 31 years prior.
Genetics, the study of genes and heredity, is a field that has been around for decades now. From the first plants being genetically modified in the 80s to the first sequencing of the human genome in 2003 to the first use of CRISPR in the 2010s, the field of genetics has become one of the premier sciences of the modern world.
Along with GMOs, CRISPR is the most widely recognizable technology to stem from this field. But what exactly is CRISPR? In order to understand CRISPR, one needs to understand the importance of what it deals with, DNA.
DNA is essentially the code that every living thing has that tells them how to sustain itself. This includes directing cell reproduction, protein synthesis, and everything else an organism needs to survive. Many would be surprised to learn that the organisms responsible for this revolutionary technology are actually our sworn biological enemies, bacteria and viruses. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. These repeats are actually repeats of bacterial DNA. In 1987, Japanese scientist Yoshizumi Ishino accidentally stumbled upon this mysterious sequence of DNA, but did not know what it was for. It was not until 13 years later that researcher Francisco Mojica and his team discovered the true purpose of the sequence.
When bacteria are attacked by a virus, it stores part of the virus's genetic code along with CRISPR DNA, allowing it to remember the strand of virus, similarly to how our immune system remembers microorganisms using antibodies. Thus, when a similar virus attacks the bacteria again, it uses a CRISPR-associated protein called Cas9 to quite literally cut the DNA of the virus, killing it. So essentially, one of the most significant scientific discoveries of the last 50 years originates from the ongoing struggles between viruses and bacteria. But how does this apply to more complex life, like humans?
Well, in 2012, researchers Jennifer Doudna, George Church, Feng Zhang, and Emmanuel Charpentier fully unlocked the potential of this sequence of DNA when they discovered that scientists can use the ability of CRISPR to cut DNA to delete genes, insert genes, or drastically modify the genes of any living organism.
In order to grasp how significant that is, we need to understand the power of the genome.
In many ways, DNA is the key to life. Thus, the ability to edit genes is literally life-changing. Because DNA is so crucial to survival and controls the production of proteins which have so many functions, when mistakes occur in the copying of DNA, called a mutation, it can be disastrous.
While many mutations are harmless, just as many can lead to devastating health problems. Genetic mutations are the direct cause for disorders such as
sickle cell, autism, Down syndrome, colon cancer, Crohn's disease, Huntington's disease, skin cancer, and many other diseases. According to the National Human Genome Research Institute, a part of the National Institutes of Health, nearly all diseases have a genetic component.
With the power to edit DNA, scientists can potentially treat or even cure a vast array of illnesses, including ones that previously had no treatment options. It is difficult to underestimate the magnitude of this potential.
Hundreds of articles have already been written about how CRISPR could potentially change the world forever.
That's not all CRISPR can do, however.
According to Dr. Harita Chevron, a molecular chemist who previously worked with Dr. Doudna, CRISPR has the ability to potentially diagnose diseases faster and more accurately than any other current method that we have due to its ability to seek out specific genes.
And those are just the medical benefits that CRISPR may offer. Many scientists are just as excited about the agricultural use of this technology. The prerequisite of every living organism casts a very wide net. As a result, the use of CRISPR will not and has not been limited to solely humans.
Plants and animals very much fit into this category, and as a result, scientists are already thinking of ways to improve the usefulness of crops and farm animals. By editing their genes, crop developers can imbue plants with more desirable traits that could allow them to live longer, resist pesticides more effectively, provide more nutrition, and other things.
While farmers have been selectively breeding domesticated animals for thousands of years, by utilizing CRISPR, animals could be completely optimized to produce as much product as possible, such as meat or milk. There are even more creative ways that CRISPR can be used. A study conducted in 2020 found that by editing the genes of male animal embryos to make them sterile and then introducing them to the stem cells of a male donor, they could create surrogates. These surrogates, nicknamed superdads, produce sperm with the genetic material of the donor animal. This is greatly beneficial to farmers, as when the superdads reproduce, their offspring will only carry the genes of the donor animal.
This allows farmers to selectively breed animals to have specific traits of other animals, producing special breeds of animals that could be optimized for farming. This technique could also be used to improve the genetic diversity of endangered species, which is a major concern for scientists.
With the synthesis of genetically modified crops and animals, the idea of worldwide food security is a possibility that could finally be realized, thanks to CRISPR. All of this sounds good. Great, in fact. It seems that with the use of CRISPR, scientists could potentially solve many of the largest problems that plague humanity today. So what could He Zhuangqi have possibly said to enrage such a large group of people regarding CRISPR? Well, while CRISPR at first seems like a flawless technology that has the ability to help millions of people, many stipulate that it has the ability to harm as well. It's time to explore the dark side of CRISPR. That fateful day in November 25th, 2018, He Zhuangqi announced the birth of the first genetically modified human embryos, two twin girls.
He proclaimed that he edited their DNA to make them resistant to HIV infection. At first, this may seem like a revolutionary breakthrough, a landmark of science that will be remembered for its significance for decades to come. Perhaps a permanent cure to the HIV pandemic could finally be realized, and this may open a door to serious treatment of other difficult diseases, such as cancer. However, contrary to what you might think, Huangqi was met with worldwide backlash and collective disdain from his peers.
Why is this, you may ask? Well, there are actually multiple reasons. First, while gene editing technology has been constantly evolving and improving, most scientists believe that it's still not accurate enough to be safely used on embryos without the risk of potentially damaging genetic mutations occurring.
Also, when the genes of an embryo are edited, the changes made to the genome are spread through their germline, meaning that their descendants can exercise the same changes in their traits. While this seems beneficial if the edit is eradicating a genetic disease, if not, it can be incredibly alarming. If a mistake is made editing one embryo, it could affect offspring for generations.
Thus, since there are other, safer alternatives for treating HIV, the risk of human embryo editing seems too great to overcome until even more advances are made in the field. Another one of the reasons is the matter of consent. Ethicists stress that consent is very important to the matter of human gene editing. However, unborn embryos cannot give consent to their genes being edited. But perhaps the most important reason why Yongqui's research was so controversial was because of its implications. While CRISPR technology has been steadily improving and developing since its debut in 2012, ethical concerns about its future have also been steadily compiling.
Imagine this. You are about to start a family. You and your spouse really want to have a child. Your friend, who is a doctor, hears about this and approaches you, saying, "How tall would you like your child to be?" At first, you are confused. Then your friend keeps going, saying, "What color eyes would you like your child to have? What color hair? If you could choose how your child acted and looked like, what would you choose?"
You are taken aback, stunned that your friend is approaching you with the idea of seemingly customizing your child. This sounds ridiculous to you now, of course, but in a relatively short amount of time, the situation might be a reality for many people. There are even more reasons why some ethicists are concerned about the future of human embryo engineering. Think about the current in vitro fertilization industry, in which couples unable to give birth through other means receive a human egg from a donor.
People often browse websites and catalogs of donors and search for donors with specific traits such as hair or eye color, race, height, and even intelligence. Prospective parents are even willing to spend much more based on the quality of the egg, that is, based on the desirable traits that the donor has. There's even been a link between higher egg prices and higher SAT scores.
This could potentially be indicative of a future human embryo industry. Those who are wealthy will have the ability to customize their children, giving them advantages in intelligence and health, while those who are less fortunate will continue to suffer from these genetic disorders that they cannot afford to treat. Thus, the socioeconomic gap between the poor and the wealthy could continue to increase. While the fact that only a select few will be able to afford CRISPR treatment may seem far-fetched, the commercialization of the current medical and agricultural industries indicates that this is very possible.
Already, companies are placing patents on CRISPR sequences and technologies, meaning that they can control the price of the use of the technology, restricting the movement that it can provide behind a paywall. This applies to agriculture too. Farmers may be denied access to new, revolutionary gene edited crops because they cannot afford it, leaving them at a disadvantage that may cost them their livelihood.
All of these future ethical concerns lead to the next logical thought. How do we prevent all this from happening? Well, in that very loaded question lies another controversial topic in the world of science: regulation.
Hey research led to widespread alarm throughout the scientific community. Many thought that this was the sign that regulation of CRISPR technology was needed. Scientists such as Jennifer Doudna, Emmanuel Charpentier, and other prominent researchers called for a moratorium or temporary collective ban on the use of CRISPR on human embryos.
However, it is not so simple. With He Jiankui as an example, many scientists are concerned that with ample resources, researchers can simply bypass the guidelines that are put in place on human genome editing and continue research on human genes. So, what can those of us who aren't legislators or scientists or bioethicists do? Well, we can petition to be more involved with the decisions that are made regarding human gene editing because this will involve us just as much as them. This is an important issue because CRISPR inevitably will affect all of us in some way in the future. And with the ability to help so many millions of people, we need to make sure that it's doing this ethically and safely without leaving many other millions of people in its wake. Thank you for watching my documentary. My name is Tomi Fularuncho, and I'm a junior at Howard High School, and I'm enrolled in the independent research course, which enabled me to study this topic and to make this documentary. So thank you, Ms. Laha, and thank you for watching.